Phospholipids can move sideways, rotate in the membrane, but moving from one layer of the membrane to the other, called flip-flop, does not happen easily and requires protein. That is where Scramblase and Flippase come in. Today, we will look at how phospholipids move within membranes, focusing on Scramblase and Flippase. Before I explain anything, tell me, what do you think they do?
I think Scramblase scrambles phospholipids.
And Flippase flips phospholipids.
Yes, that is actually accurate. Well, let's not end there. An enzyme bound to the cytosolic surface of the endoplasmic reticulum synthesizes phospholipids from free fatty acids. These newly synthesized phospholipids are added only to the cytosolic monolayer of the ER lipid bilayer.
Wait, won't the phospholipid just overload the cytosolic layer?
How would the non-cytosolic monolayer even grow? That feels unfair.
And that is exactly when we need our hero, Scramblase. Scramblase is a transmembrane protein that catalyzes the transfer of phospholipids from one monolayer to another. In the endoplasmic reticulum, Scramblase transfers some of the newly synthesized phospholipids from the cytosolic monolayer to the non-cytosolic monolayer. This ensures symmetric growth of both monolayers of the lipid bilayer. In the endoplasmic reticulum, it helps the non-cytosolic monolayer keep up with the growth of the cytosolic monolayer. As a result, phospholipids are randomly distributed in the ER membrane, regardless of the type of phospholipids. Portions of membrane pinch off from the endoplasmic reticulum to supply fresh membrane to other intracellular compartments, such as the Golgi apparatus. These membrane vesicles then fuse with the Golgi apparatus membrane. Flippase is a transmembrane protein present in the membranes of the Golgi apparatus and other cellular membranes. Flippase catalyzes the transfer of specific phospholipids, not random ones, to the cytosolic monolayer. Flippase transfers specific phospholipids, such as phosphatidylserine, to the cytosolic monolayer, maintaining the presence of certain phospholipids only in a specific monolayer.
So, in simpler terms, Flippase makes sure certain phospholipids stay in certain monolayers.
Yes, and this maintained phospholipid asymmetry is essential for normal membrane function and proper cellular activity. Even when a transport vesicle is pinched off from the Golgi apparatus and later fuses with the plasma membrane, lipid asymmetry is preserved.
So, the phospholipids keep their orientation.
Exactly. Specific phospholipids maintain the same orientation and remain in the same monolayer after vesicle transport.
You know what?
Whoever named these two proteins,
Should get a Nobel Prize in naming.
what a genius!